I'm Dr. Cody Strom, and I'm lucky to be joined with some colleagues of mine from our mutual study group, Dr. Bree Wisseman and Dr. Alex Claiborne. So we've worked together under Dr. Linda May, and we've done different aspects of research, and particularly in the exercise and pregnancy world. So our topic that we're going to talk about today is prevention through intervention, the growing science of maternal exercise and how it improves the health of mother and baby. So this study that the three of us worked on or are continuing to work on actually comes from our mentor, Dr. Linda May. So I worked under Dr. Linda May when it was still the enhanced study from 2017 to 2020. And then Dr. Bree Wisseman worked with the May Lab as it transitioned from the enhanced study to active mom study. And then Dr. Claiborne came in as her postdoctoral researcher and is still currently working with her today. So yes, there we go. So a little bit about me and where I came from prior to where I'm at now. I grew up in Grand Rapids, Minnesota, and yes, like all good Minnesota individuals, I played hockey, and I also played tennis. And so I had this love of sports and exercise and health. And once I didn't get drafted to the NHL, I didn't really know what to do. So I ended up going to college and like anyone else does and trying to find themselves. And I went to University of Minnesota State Mankato. And I actually got a degree in both biomedical science and exercise science because I really couldn't decide. I love the human body and the medical side of things, but I still had that athlete in me and wanted to learn more about exercise. And during my last semester, I needed a couple credits to fill, and I ended up getting into some research. And that actually led me into getting my master's in exercise physiology at the University of Mankato. So during my time in Mankato, I worked on multiple research projects diving into different aspects. So one of them, I did body composition, validating a new method using bottle buoyancy. I looked at, the critical power model, critical speed model, and how, the difference with aerobic and anaerobic athletes, that aerobic versus anaerobic power, and depleting those substrates. And so that was a really cool study. Got to go over to South Africa for that. And then my thesis was actually in validating a customized submax protocol. So I really dove into some metabolic aspects during my master's program, and it was really great experience. And later on, I mean, during my experience there, I knew I wasn't quite done yet. And so I started looking towards a PhD. I was still thirsty for that knowledge, and luckily, I got recruited to East Carolina University. And this is kind of the funny story that I was talking about with Dr. Wisseman and Dr. Claiborne. So you're going to learn that you'll make some mistakes along the way. And one of their mistakes they mentioned, maybe not changing a college name on an application. But I sent a mass email out to the professors at ECU saying, "Hey, I'm looking for a doctoral spot or a PhD spot." You should always address emails to single people and not mass departments. But luckily enough, I pretty much was ghosted and I didn't have I didn't think I was going to end up at ECU, but it wasn't until the fall that Dr. Linda May actually did reach out to me. So she got my name from one of her colleagues, and she reached out and, she told me about her study, and that's kind of how I got to East Carolina. So you never know, sometimes even when you make those little mistakes, I mean, even just putting yourself out there can really lead to these big things. And so she really talked to me about this exercise in pregnancy and how the effects of maternal exercise intervention and nutrition can have positive outcomes. We kind of think of the negative outcomes of smoking and drinking, but now we want to look at positive effects. So going into a little background of that, so we've seen the rise in childhood obesity, and it really is kind of correlated to that maternal obesity. So We see these obesity risks as well that lead to increased cholesterol, triglycerides, risk of heart disease, which is the leading cause of death, increase in type 2 diabetes. So we try to look at when the earliest intervention point is. And Dr. Linda May worked under a few individuals such as Dr. Clapp, who did a lot of research in this intervention point. And the earliest intervention isn't during your lifespan, but it's right in utero. So we can actually have this, what the title of this presentation is called, a prevention through it, or prevention through intervention. So, and it's based off this fetal programming done by David Barker. So he looked at birth records with cardiovascular disease and saw, with the modifiable risk factors, like I mentioned, the smoking, drinking, but we have this positive of the diet and exercise. We know that maternal nutrition can lead to different adaptations post utero, so postnatal. So undernutrition kind of anticipates a nutrient poor environment. Overnutrition, it stores that excess fat, especially as visceral fat, which is bad fat. And we have this change through pregnancy with the energy demands. We have this metabolic switching, which will kind of throw glucose utilization and fat storage in different aspects. So first trimester, we increase. Second to third, increase in the lipids, decrease that glucose utilization. So there's a lot of things that are occurring through pregnancy. And so we want to really try to implement these positive effects. So we've seen lipids during pregnancy that they are important. Cholesterol for cell membrane, steroid hormone, CNS development, triglycerides being used as a fuel source. But we can also see that elevated lipids have both negative maternal outcomes and negative infant outcomes as well, with preeclampsia, gestational diabetes, preterm birth, increase birth weight, fatty aortic streaks. So these are streaks on the heart. That hypercholesteremia is going to increase those, again, that total cholesterol with those fatty aortic streaks, leading to more low density lipoproteins. So increased monocyte recruitment, and that's just early atherosclerotic lesions. So having that high cholesterol can really put the infant at a disadvantage even before they're born. Um, so and then we talk about the infant body composition. Matola said it best that obesity begets obesity, as in it's this cyclical nature that if mother's obese, then they tend to have obese babies. They continue to propagate. And it leads to those large gestational age, more fat mass. We also see that small for gestational age can increase the risk of obesity and diabetes as well. So we kind of have what's called this U-shaped curve that we want to try and get right in the middle there, because that's going to decrease the odds ratio of developing, obesity and diabetes later in life. And so the birth adiposity is strongly predictive of that at 5 months. And at birth, decreased childhood abdominal fat is shown to decrease the risk of obesity and decrease the risk of diabetes. So we kind of done this intervention now, and Dr. May started this intervention to help these mothers and help the infants so this enhanced timeline, we actually take in these pregnant women at they're at the end of their first trimester between the 13 and 16 weeks gestation, and we get them enrolled. And this is the original timeline from Dr. May, and they would do pre-measures. We would actually have maternal measures at every 4 weeks, 36 weeks, taking some fetal measures, and then we have like our final measures as well at 36. Once they deliver, we get birth measures, and then we'd also look at infant measures at 1, 6, and 12 months, and she's even continued on in a different study to look at up to 13 years. So the traditional intervention when I came in was 3 supervised exercise sessions per week, moderate intensity, 50-minute sessions, and exercise dose was calculated weekly and averaged over that intervention. Now that really hasn't changed too much, except for that we've actually added modes now. But going into some of the research that I started with, in the initial project with Enhance, I actually started looking at cholesterol. I helped a little bit with this initial paper, and we saw this correlation with exercise dose with pre and intra-pregnancy fetal measures as it pertained to some of these lipid levels. And so it got me interested in lipids. And so I really wanted to look at other dietary things, particularly the omega fatty acids. So polyunsaturated fatty acids, which we know we have our omega-3s and our omega-6s. And we always tell pregnant women that they should have some kind of supplement for DHA and EPA. And those are the ones of focus because these polyunsaturated fatty acids are super important for the structural components of the cells. They're precursors for eicosanoids making prostaglandins. They can't be synthesized. And we want to have a 4-to-1 ratio of the omega-6 to omega-3s or 1-to-1 ratio. But our diets are really high in that. And so the importance of the omega-3s, you'll see here, is that we are derived from plants and fish. They compete for the enzymatic systems, but they're less potent inflammatory eicosanoids and they're more anti-inflammatory. And we can actually increase the placental fatty acid transport proteins, which is good. It's getting more energy to the fetus and as well as, not causing inflammation. And then the DHA, really important for fetal brain development and retinal development as well. But then we also have these omega-6s here, which are pro-inflammatory. And I won't bother you too much with the biosynthesis pathways, but they share these same enzymatic pathways. And really, what it comes down to is what kind of eicosanoids are being made, and they're going to differ in their strength of inflammation if they're pro-inflammatory, because even omega-3s are pro-inflammatory. But there are some that are anti-inflammatory, whereas the omega-6s are all inflammatory and they're very high inflammatory. So to sum up, omega-6s and versus omega-3s, omega-6s, everyone panic, whereas omega-3s, you need to relax. So I found it important to look at these PUFAs, and I did. I saw these correlations in the maternal environment and what it can do, decreasing the inflammation, oxidative stress, free radicals, improving maternal lipids, increasing lean mass, fat mass, nervous system development, and then the recommended amount coming from here, seeing that, with different kinds of consumptions, we could see the decrease in triglycerides, increasing in good cholesterol or HDL, and even with supplemental trials, again, decreasing those triglycerides, increasing HDL. So we also saw with the infant body composition we talked about before, is that with the DHA and EPA supplementation, increasing gestational period, which is good. They're going to term, increasing birth weight and length, lean body mass, which is important, decreased fat mass at birth in 6 weeks, increased growth rate for preterm infants, and then continuing that trend later in life. So these are studies that were done by others, but I wanted to add this to Enhance. So supplementing into Enhance, I went and trained in the mass spec lab. So this is actually screening in cord tissue, opioid and stimulant exposure targeting with umbilical cords. Now, the main part about this was learning the mass spec. So this is a very high intense tool to quantify metabolites. And so if I wanted to look at DHA and EPA, I needed to learn this equipment, and then this could actually measure in very, very small, precise amounts. So I could measure down to the microgram. And so Dr. Linda May, under her study and her guidance and mentorship, allowed me to add this portion into her study. So during the PUFA analysis, I jumped in on the 16 and 36 weeks. I implemented fasted blood draws and made sure that they were done in the morning. And we made sure to store at the -80 Celsius. And then I also added different PUFA analysis, or excuse me, different cholesterol analysis using a CholesTech. So we got real-time cholesterol that we actually now use even for the infants. So doubling that, I'm able to look at both the exercise, the PUFAs, and our measures of outcome. So the intervention timeline that I followed was that, again, I did our blood draws, we went through the intervention, and then I got my first measurement and first study with my maternal lipids and PUFAs at the 36 week. And then I transitioned into infant body composition. So what we ended up seeing was that through my study that decrease in the triglycerides with increased exercise dose. So really what it came down to was that exercise was uh, much more of a stronger correlator to the decreasing triglycerides and a much stronger factor in decreasing triglycerides than the DHA and the EPA were. So this can help, the negative effects of the maternal and infant outcomes associated with that hypertriglyceridemia. So it's not to say that the DHA and EPA weren't important. They did help, but we saw significant correlations with exercise. So it just shows how important physical activity can be. So infant body composition was the other measure that I focused on with the DHA and the EPA. And once again, we found that same kind of trend where it was really the exercise that was a stronger indicator of decreasing the body fat, and that stronger correlation. So it seems that exercise is a little more important and can really make up for a lot of that, maybe some dietary issues. So it is really important to stay active through pregnancy, and I think that really helps regulate metabolically. And so those were the two main outcomes that I got through the enhanced study and what I was able to add to within that study. And then it progressed me into getting a assistant professorship later on, teaching courses I love to teach with kinesiology, physio, exercise, biomechanics, working on different committees, and using my knowledge with the bioenergetics and the different metabolites and looking at the metabolic profiles of mothers. I've now, in addition to the May Lab studies, have reached out to our community partners working with different hospitals to actually look at a resistance band study in pre-diabetics for the underserved communities using a home-based model. So that is my experience with the enhanced study. And so once I got done in 2020 and got my professorship, it was Dr. Bree Wisseman that ended up taking over and becoming Dr. May's new PhD student. And with that, I'll let her take over.
Well, thank you so much, Dr. Strom. As previously mentioned, my name is Dr. Bree Wisseman. I am at the University of Rhode Island as an assistant teaching professor, currently teaching exercise testing and prescription and intro to exercise science. A little bit about me and my background. I am from a small town just a little bit outside of Washington, D.C. called Culpeper, Virginia. After I graduated from high school, I made my way over to Harrisonburg, Virginia, where I went to James Madison University and pursued my bachelor's degree, focused in kinesiology from the start, with a minor in coaching. And when I went into undergrad, I really knew two things. I really knew that I liked being physically active. I was a athlete so far throughout my career, in high school and going into college. And I knew where I wanted to go. I wanted it to be doing something that could combine that interest with helping people. And so, like, I think a lot of kinesiology majors go to, the best way I thought to do that was through physical therapy. And so for the first 3 years in undergrad, I was a pre-physical therapy minor. And during that time, I got a lot of experience working and shadowing in physical therapy clinics. I was a patient in a clinic for a long time during that process. And throughout all of that, I realized that physical therapy really wasn't for me. And so, but I still wanted to combine my interests in enjoying physical activity and what I was learning through exercise science and being able to help people. And so from a friend's recommendation, I actually signed up for an internship with the Human Performance Lab at James Madison University and absolutely loved it. So I was working as an undergraduate research assistant for a study that looked at the influence of moderate exercise training on heart rate variability. And through this experience, I later got the opportunity to give my first poster presentation at a conference, as well as my first author publication, as shown here. And so from that experience, I loved every second of it. And what I realized was that this was just a different way of being able to combine my two passions of participating and doing physical activity and my love for being active and helping people, but just in a different way. And so because I really loved the research process and my time in lab, and I was loving everything I was learning in my major classes for exercise science, that told me that it was time to pursue my master's degree at the end of my bachelor's. And so I stayed at James Madison University and pursued my master's, graduating in 2020 with a concentration in exercise physiology. And during my master's, I worked on my thesis project that was looking at how different fatty acid compositions in a high-fat meal impacted postprandial airway inflammation, as well as some different blood markers. And so for this particular study, this is actually great. Dr. Strom went over the difference between omega-3s and omega-6s, so thank you for that. My study, we were having individuals, college-age individuals, come in, and on 3 separate occasions, they'd get 3 separate smoothies, and those smoothies would be either high in saturated fat, high in omega-6s, to omega-3s, like that Western diet ratio that Dr. Strom introduced earlier. And then the third condition was a high omega-3 to omega-6 condition. And we were just looking at how this impacted airway inflammation and different markers in the blood. And so for my thesis project, we did get a publication out of it looking at that postprandial airway response. And what we found was between the 3 conditions, it didn't really alter or influence what happened to airway inflammation over a 4-hour period post-consumption. But what was interesting, and for our second publication from this study, was that even though we didn't see any differences in airway inflammation, we did see that through metabolic responses measured with triglyceride content and glucose content in the blood, we could get an idea of what the metabolic load was. And metabolic load can be a really good indicator of what the whole meal is driving, as opposed to just the high fat content or the fat type. And so what we found was that while for triglycerides, we didn't really see any significant difference between the different types of smoothies. When we looked at glucose, we did see a significant difference between the groups across the 4-hour time period, depending on the type of smoothie that you got. And so the saturated fat smoothie had a much higher glucose response in the blood compared to the omega-3 fatty acid smoothie. And then we could combine the triglyceride and the glucose measures to get this idea of MLI, which is the metabolic load index. And so by combining these, we're looking at, well, what is this? What is the food content really driving in the body, and how is that impacting our metabolism as a whole? And so through this MLI, we also saw that the smoothie that was high in saturated fat had a much larger increase in this metabolic load at that 2-hour time point compared to our omega-3 fatty acid. And so during my master's, through this thesis experience, I was immediately hooked. I loved the research process. I really enjoyed my time doing the dietary sort of analysis related to health markers. And I learned a lot about inflammation in general, not just related to the airway, but to the whole body, and how inflammation in the blood can really drive a lot of these changes. And so I knew I wasn't done yet. I knew I wanted to pursue my PhD. And so when I was going into a doctorate program, I knew 3 things. I wanted to continue exploring what my interest in the role of inflammation has on health was. I wanted to sort of shift my personal focus away from the dietary aspect and more focus on the effect exercise had. And then I wanted to learn more about female exercise physiology. Outside of my thesis, I learned during my master's, through specific coursework, that there were a lot of differences when it came to exercise physiology between males and females. And I guess so far through my schooling, I just hadn't considered that the physiology we were talking about full time didn't necessarily apply to me and my own physiology. And so this particular study by Emma Cowley, that was published in 2021, did a really great job at looking at the sex gap in sport and exercise science research. To kind of sum this up, they found, looking at 6 major sports science journals, that of the studies that were published between 2014 and 2020, only 34% of those studies that were published included a population that had females in it, whereas only 6% of the publications exclusively looked at females. And so I really wanted to sort of target female exercise physiology going into my doctoral work. And this graphic from Corson that was published in 2024 sort of does a nice visual depiction of this volume of knowledge in terms of exercise physiology. And so we know a lot about how males respond to different exercises related and how their exercise physiology works. We know a lot less when it comes to females. And when we start looking at pregnant individuals, we know even less than what we know. And so that led me down to Greenville, North Carolina, to study with Dr. Linda May in her lab at East Carolina University, where I pursued my PhD in bioenergetics and exercise science. And so going into Dr. May's research lab, it was an incredible opportunity, and I'm so glad I got the chance to work with them and the amazing work that they do. And so kind of coming into it, I had this idea of, well, how can I help progress our research group as my time there, but also sort of target this towards what my specific interests were, right? And so to remind you, I was really interested in focusing on that exercise effect. And Dr. May's group does a fantastic job because they have a bunch of different exercise modes going on to sort of focus this back in. So the first way that I did this coming into Dr. Linda May's research group is looking at in-person versus at-home exercise. So I started my PhD in 2020, and so it was really in the middle of the COVID-19 pandemic. And so one way to sort of start looking at exercise again for me was just to see, well, are there differences between individuals who work out in person, who are coming into the gym and meeting with us one-on-one, versus the individuals who are choosing to do at-home exercise sessions and report back in. Because of the COVID-19 pandemic, at the time we were able to do a little bit of both. And so that led to this paper, my first paper with Dr. Linda May's group that looked at maternal cardiovascular and birth outcome responses to at-home versus in-person exercise during pregnancy during COVID-19. And what we found in this related to the maternal cardiovascular system is that individuals who were exercising at home had a larger increase in late pregnancy in their systolic blood pressure, whereas in-person exercisers actually saw a slight decrease in systolic blood pressure, whereas diastolic blood pressure for both groups increased slightly, but it increased to a much greater extent in our at-home exercisers compared to our in-person exercisers. And so that sort of piqued my interest on, well, how does exercise, how does this intervention type really drive changes? And because, as COVID-19 sort of wound down, we were able to start bringing all of our participants back full-time in person, which was great. And so this then drove me to start being interested more in exercise mode. If you did cardio-based activity versus weightlifting versus a combination of the two of them for your exercise programming, does that impact your overall outcomes? And then I was still had in the back of my head this idea and this interest in the role inflammation plays on overall health outcomes. And so to give you a little bit of background, when it comes to pregnancy inflammation and the impact of health, we know that pregnancy in general is an inflammatory event. However, what happens when we get certain conditions related to pregnancy is that it can cause a sort of imbalance to this pro and anti-inflammatory shift that we should see. And so to put it in perspective, there's about 20.1% of pregnancies that are affected by gestational diabetes, 4% that are affected by preeclampsia, and 29% that are affected by pre-pregnancy obesity. And all of these conditions are associated with excessively high levels of pro-inflammatory biomarkers like TNF-alpha, IL-6, IL-1 beta, and CRP, with low levels of anti-inflammatory biomarkers like your IL-10. And so, what I was interested, considering that there's this tie between inflammation and pregnancy, was well, how does this impact mom's health? And it's not just mom we're concerned about anymore. And so, to kind of outline what's happening, is when mom has these high levels of pro inflammatory biomarkers, it increases her risks for certain adverse pregnancy conditions like gestational diabetes, preeclampsia. But that high level of pro-inflammatory biomarkers can also be translated and result in high levels of these biomarkers in the placental tissue, which can then enter fetal circulation through the umbilical cord. And so it's exposing baby to these higher levels of pro-inflammatory biomarkers in utero. And so, to sort of tie my interests together, what I was interested in looking at was, well, what exists when it comes to exercise mode or exercise type and the influence on inflammation? And so we know in non-pregnant individuals that aerobic exercise or cardio-based exercise and weightlifting exercise, when they're done independently, are both good at decreasing these pro-inflammatory biomarkers. But when we combine resistance and aerobic exercise into our program, we actually get a much greater decrease in these biomarkers. And so I was curious to see, well, does this translate into pregnancy? Does this happen with mom? Can we help control the level of biomarkers to a safer level so that we're helping mom, placenta, and baby out? And to sort of summarize the literature that's out there, there's a lot left unexplored when I started this project. And so in terms of aerobic exercise, Dr. Clapp did a lot of research in this area. He did see that when you are doing aerobic exercise alone, you actually see a decrease in these biomarkers. And then Acosta Manzano did another study looking at combination exercisers. Again, in maternal blood, you're seeing these lower levels of pro-inflammatory biomarkers. And Acosta Manzano also looked at that effect in cord blood and saw similar findings for certain biomarkers. But there's still a lot left unknown. And so that led me to my question related to Dr. Linda May's project was, does exercise mode during pregnancy differentially impact inflammatory biomarkers? And so, to sort of show an abridged version of where my project kind of got my data from, looking at that overall timeline that is pretty similar to the Enhanced by Mom study that Dr. Strom presented earlier on, and Dr. May's new study, the Active Mom studies, we recruited individuals still before that 16-week mark, but now we could recruit them even earlier than the 13 weeks initially shown. And during that time, we still did our enrollment measures. And so we did pre-exercise testing. That's where we got our first fasting blood draw. Thank you to Dr. Strom for introducing that to the study. And then they came in, we randomized them after that enrollment measures to one of the 4 exercising groups, being aerobic, resistance, combination, or control. And those exercising groups still follow the similar protocol to what Dr. Strum presented earlier on, where it's still 3 times a week, 50 minutes coming in, doing activities related to those exercising groups. At 36 weeks, we got our post measurement for mom so that we could look at that effect of exercise on mom. And then at delivery, we collected the placenta, tissue, and cord blood so that we could continue looking at this communication and inflammation as well. And also, as a thank you to Dr. Strum for introducing the metabolomics to Dr. Linda May's research study, seeing some of the outcomes that he got from that metabolomic search and targeting DHA and EPA specifically, I was really curious to see if, well, what if I don't want to target one specific thing? What if I want to see everything that changes as it relates to this question, because there's so little known? And so for my project, I did global proteomics to see if there is any differences in protein abundance that related to inflammatory pathways during pregnancy dependent on this exercise mode. And so similar to the metabolomics that was introduced earlier, global proteomics, we had a sample prep kit that you see up in the top right, and we ran that on an Orbitrap mass spectrometer so that we could start looking at all of the differences in protein abundances across our groups. And so in our placenta tissue, we found that for our aerobic resistance and combination groups, there were a difference in total uh, a difference in total proteins that differed in abundance compared to the control groups for those exercisers. And then when we looked specifically at the ones that were significantly different in abundance, we actually saw a differing amount that were related to inflammation or immunity. And so for the second part of my project, we wanted to look at, well, how does this impact overall pathways? And so we were able to do that looking at the cord blood. And not only did we see differences in protein abundance, and it was different between all of the exercising groups, but we also found specific pathways that related to inflammation and immunity that were decreased in our exercisers versus the control group. And so for aerobic, there were 4 pathways identified versus control. Resistance, there were 3 pathways identified versus control. And combination, there were 7 pathways versus control. And what is so cool related to this question is that for the combination exercisers, of those 7 pathways, 2 of them were the same pathways that happened in the aerobic exercise. 3 of them were the same pathways that happened for the resistance exercisers. And then there were 2 unique pathways that were specific to our combination exercisers. And so we started seeing this difference emerge in what's happening, all based on exercise mode, outside of just aerobic exercise. And then to sort of tie it all together, we wanted to see it's not always exercise type that drives these changes, but we started looking at more regression analyses to see, well, how do our other exercise metrics or the FIT principle impact these markers as well? And what we found is that exercise frequency and duration, or F and T, was able to predict some of the maternal inflammatory biomarker outcome. Exercise intensity was able to predict some of the placental tissue inflammatory biomarkers, and exercise volume and mode were predictors of some of the other ones. And so overall, this was suggesting that there are multiple exercise metrics that may influence outcomes. And so from this point forward, I will pass it over to Dr. Claiborne to talk a little bit more about this idea of the FIT VP principle and how his research with the project has carried on.
Thanks everyone for having me. I'm Dr. Alex Claiborne. Thank you to Dr. Strom and Dr. Wisseman for sharing and doing so much of the elemental work that led to much of my opportunities in Dr. May's lab. I'm going to start by introducing myself and a bit of my background and how I ended up where I am. I'm now a third-year postdoc in Dr. Linda May's lab. My name's Alex Claiborne, 32 years old right now. I'm originally from Winston-Salem, North Carolina, so about 3 hours up the road from where I am currently. And I have completed 6 years of post-baccalaureate work. There have been a lot of fun moments alongside of that. So if you're going on to grad school and whatnot, it's a lot of work, but you have a good time and meet a lot of good people along the way. And then I've spent the past 27 years or so biking with my friends and a lot of fun adventures and bruises associated with that as well. So to start off, biking's very core to me. Like that's something I've done ever since I was a kid. So naturally, when I graduated with my doctorate, I of course brought out one of my favorite bikes and took a picture with it. But the in between, I plan on giving you a little overview of today, and hopefully that helps explain like where I've ended up, because I've always kind of tried to follow things I'm interested in work as well as outside. So my background is I began my sort of formal schooling at UNC Wilmington in North Carolina. I got my bachelor's in biology in 2015. Started as pre-med, actually switched interests though while I was there. I went on to graduate school, master's program in kinesiology at Miami University in Ohio, which I received 3 years later in 2018. And then moved to Ball State University in Indiana, where I got my PhD in human bioenergetics in 2022. That's now 2 years ago. But I always sort of joke about this. Most of my education in exercise physiology came from sore legs and tired lungs on long bike rides with my friends. And there are some pictures of some of my buddies from Indiana. So let's start with my bachelor's study. As a college student in Wilmington, I was a bookworm. Obviously, it was a fun place to go to college as well. And the sun was always shining, so I was always on my bike. And to maintain a happy attitude and smiling face all the time and an energetic personality, I'll give you a second to guess what the question mark is hiding. And that would be a strong cup of coffee. So initially, my exercise physiology interests sort of stemmed from while I was a student in biology, we were learning about physiology, and I was interested in how caffeine kind of influenced exercise performance in a positive way. So presented a paper on that in a senior year seminar, and the rest was history. I quickly switched from pre-med to sort of following my passion and in exercise physiology. Now, before going on to grad school, you might have noticed I had a gap year before my master's began because exercise physiology sort of is this applied science that takes sort of biological knowledge and applies it to an exercise session or training. So I was missing some prerequisites in talking just about the human physiological response to exercise. So I had a bachelor's in biology, but I did not have even the most basic understanding of how to apply that knowledge during an exercise session. So I actually went to I lived at home for a year, worked in a bike shop, and got my prerequisites filled at Winston-Salem State University. I took ex-phys and I took biomechanics there. And then I sort of followed where I had the most experience already between the two concentrations. Exercise physiology had a lot to do with molecular biology. However, biomechanics did not. So you guess which one I concentrated on during my master's, went into physiology, and I did that at Miami University in Ohio. And I had a lot of good times there. I'll get to this a little bit later, but my application to Miami would not have happened if I didn't have a good friend recommend the department to me. So I just want to take time to note that. When I got to Miami, I was assigned the mentor, my mentor, Helene Alessio, who I speak with to this day. And there's a picture of Helene. My project revolved around heart rate variability mostly. Dr. Alessio gave me a little 2-lead electrode set, and I hit the ground running with it, developed a few different projects, which I'll go over in the next slide. But basically, I was focused on physiological stress and how heart rate variability would respond to it. And that was in healthy college students at the time. And there's a picture of me presenting some of my data at an ACSM conference. So I looked at collegiate athletes. I was interested in whether heart rate variability correlated with a lot of the body composition and blood pressure measurements we would get through a program at Miami, and found some correlates. We actually published this data just suggesting that heart rate variability is one of these sort of correlating indicators of cardiometabolic health. I also looked at the response of heart rate variability before and after acute exercise in some competitive cyclists, i.e., those were my friends. So this was very much a convenient sample, and found that the trained cyclists, obviously responded a little bit differently from the healthy but non-trained controls. And then finally, potentially the most exciting part, I actually was awarded the opportunity to travel abroad to Nepal with into the Himalayas with a group from Miami. And we took our heart rate variability little 2 electrode device over and set it up in some of the hotels we stayed in, and collected data at altitude. And besides this one very obvious outlier, we found that the heart rate variability would go down on exposure to altitude, but after a period of 1 or 2 weeks, we would see some acclimatization, and heart rate variability would slowly begin to come back up. So that was a really neat experience, and I'd go on in my PhD to learn a lot more about altitude physiology, and that's something I would like to study more in the future, but haven't really gotten too deep into it so far. Moving on, so my PhD, I sort of applied to schools all across the board, in the Midwest, that is. And one of those schools was Ball State University. Shared some common interests with their lab up there. They had a pretty well storied history of just doing like basic athletic sort of physiology testing. And then there was the bit where I spent half of my interview there planning a bike ride with my to-be mentors. So once that connection grew, or occurred, I kind of knew that was going to be the place. I mean, I don't know, that was elemental. Here we are doing a bike ride later that summer. I also noticed there was a NASA poster. The lab had done some work with NASA, and I had been a plane dork since I was a kid. So all the green lights were kind of shining in my face when I interviewed and toured there. It just felt like a good fit at the end of the day. And so I went on there to study human bioenergetics for my doctorate. My mentor there was Todd Trappy. That's a picture of Todd there. And the project was actually, we looked at, it was loosely associated with NASA. I got to analyze some data from a bed rest study, where the participants would exercise in the supine position, and mitigate some of the loss of skeletal muscle tissue and power and strength that happens when they were in bed for 70 days. It's hard to believe. That model is used to assimilate the effects of microgravity on skeletal muscle, which is why they were paid well to stay in bed for so long. That data just got recently published. We focus on the leg because the leg's especially responsive to bed rest. And we saw that exercise in these individuals was protecting some of the against some of the muscle loss in the quads, and less of the muscle loss in the calf, just briefly. So I knew here's a very clear example of the positive effects of exercise. I kind of wanted to stick in that world. Dug a bit deeper into the data, and realized that actually there were metrics beyond just whether they trained or not. So the amount of training per week they were doing was actually influencing the benefit that they saw. And this is where this idea of FitV, the FIT principles, kind of originated. Because we all exercise, but we all exercise at different levels and stuff. And I'm a numbers guy, so I want to see minutes and intensity and stuff. when someone tells me exercise is good, I ask how much. But yeah, anyways, that's where that originated. We know exercise sort of, maybe this is a bold statement, but it helps a lot of things as far as health and performance are concerned. But where do we start with that? Like, here is my training volume just throughout the year. Like, should I focus more on these low volume weeks? Should I focus on the high volume weeks? Like, and with these big undulations, am I like, maybe during a high exercise week, am I less at risk of cardiometabolic disease just during that week, or how's it all work? There's a lot to be determined still, I would say. So I wanted to further study this dose response after I graduated. I wanted writing experience, I wanted lab experience, and I've been lucky enough to get all of those at ECU. The last part, I wanted a comprehensible study design. And so when the idea of studying exercise during pregnancy was pitched to me, in my interview with Dr. May, I went and looked at some of the research, and well, pregnancy is basically like exercise 24/7 already. A lot of the physiological responses to pregnancy kind of mimic what exercise does. And so I really jived well with the study design she had going. There were easy questions to answer. I wasn't a genius in DNA methylation and all that stuff yet. I could understand Dr. May's work at the end of the day, and that's what the big draw was. So I moved to East Carolina University in September of 2022 to start my postdoc with Linda May. There's her picture. My tissue processing mentor while I was here was going to be Bree Wisseman, who you might have heard in the beginning of this presentation. And of course, my statistical advisor, particularly in quadratic modeling and things of that nature, is none other than Dr. Strom. So these two helped me a ton when I was getting started here, and to this day help me write and review papers and data all the time. My project, given that I wanted to focus on exercise dose and I wanted some lab experience, would focus on the effect of dose on infant adiposity. And it had this lab element where we actually grew fat cells off the umbilical cord and tested their lipid storage. Previous to all of this work, preceding all of this work, I had done a literature review just to see what was out there on exercise dose. And I won't get deep into this, but there was some variety in the doses that had been tested already. The exercise volume per week was different, and we found it to be influential, and that basically gave us the green light to go ahead and study this. From that, I published, after that review, I published a couple sort of low-hanging fruit papers where we looked at some of the birth data, both in term and preterm pregnancies, and wanted to see if the dose was influencing any of this. And especially in the preterm, we saw that with more weekly exercise duration, the occurrence of late preterm births was increased. So we thought that more exercise per week was actually leading to a delay in delivery for preterm moms. And that delay in delivery was then also associated with a trend for increased birth weight. So these were sort of my initial findings in the FitV world, and then I got my hands dirty in lab. Well, not dirty, but you get what I'm saying. And we began our work on the infant adiposity project. And so on the left of this picture, you'll see our little adipocytes, our pre-adipocytes. We've got fat cells that we basically differentiated from stem cells, which we harnessed from the umbilical cord at delivery. And this is a really neat model because it shows us cellular changes without external influence on the offspring. And there on the right, some of the myogenic cells, which some of my colleagues here have dove deeper into. And our findings were that in the exercise group, the opposite of what we expected to see occurred. So we actually saw more lipid content in the exercisers, and this was really puzzling. I'm going to say, going forward in your science career, keep an open mind. Stick to your hypotheses, but don't expect them to work out all the time. So we wanted to chase this down because this was weird to us. We knew that infants came out usually with less adiposity if mother exercised, but we were seeing the opposite at the cellular level. And so we went and ran here's the body fat data for that, so lower in the exercisers, more lipid storage, however, at the cellular level in the exercisers. And what we saw was, while the exercise cells were storing more lipid, they were also oxidizing fatty acids more completely, basically. The complete oxidation to CO2 was higher, and the incomplete oxidation was lower in the exercisers, thus suggesting that while the lipid content is higher in these cells, the oxidation rate is higher, and I guess over that first month of life, this sort of could lead to lower body fat at 1 month and beyond. Okay. So this was the main project that a lot of this data has been coming out over the past year, and there's more to come. But yeah, that was the sort of main cellular work that I completed at lab, and we're also focused on some other areas, including maternal lactate in predicting disease, and also some of this validation of the exercise intensities that moms are performing. And with that, I'll hop into future directions. So we want to get into a further interpretation of the FIT-V findings. So why is frequency or intensity, why are those specific markers metrics influencing biomarkers that we see? And this is just a wide open question. It's stuff that Dr. Strom and Dr. Wisseman have begun to parse into with their study of molecular biomarkers in response to altered frequency and volume and things of that nature. But more work to be done there. We also have a sort of a smaller project going on validating the intensity of exercise in our pregnant women. Through this, we use a portable metabolic machine to analyze their VO2 while they're actually exercising. And from that, we can get a real-time estimate of their METs and compare that to the compendium, which has been published for years now. Additionally, it would be neat, although it will require a lot of analysis, to actually put some numbers, some more specific numbers behind exercise volume. So looking at how much weight is lifted, how fast moms are walking or running, and how hard they're pedaling when they're on the bikes, and to start to get a little bit more specific with some of this data, to drive more specific recommendations across the board in pregnant women. I also, while this is a wild dream of mine, it would be neat to accomplish some case studies in athlete women Marathon running is becoming more popular, and maybe you guys have seen some of the Olympic track athletes and other disciplines competing while they're pregnant. That's just a gold mine for me. That would be very interesting to look at just from a sort of extreme exercise training perspective. And of course, I'm sure there'll be countless other ideas in the future. So my advice, if you're going through all of this, make your own way. Be creative about it. Follow your own path as you navigate through academics and everything. If you need some guidance, a couple things that have helped me along the way is picturing myself as an adult at work, investing time in my interests, actively seeking new interests. Finding a way to merge your hobbies with your work, so taking things that you're interested in and studying them, trusting your feelings, being patient, and of course, making good friends and listening to them when they try to help. And of course, taking pictures along the way. And with that, I believe that's everything. Dr. Strom, pass the mic back to you.
Thank you, Dr. Claiborne, for talking about really where the study's at now, and then also Dr. Wisseman with, the additions and everything. And I think, just really summing up a lot of this, and you guys can add in, is, you always want to, like Alex just said, or Dr. Claiborne just said, is, finding a way to merge your hobbies with work. We all kind of have the same interests, but different interests. I'm a hockey player from northern Minnesota. Bree was coaching back in Virginia, right? Yep. And then, and Alex, you're a cyclist. Very different backgrounds coming in. And then we all kind of found this exercise physiology and this field, and we're able to learn more about the body and really relate it to our hobbies, like you said. And I think, again, being able to then transition into the May lab. And I want to, again, give props to Dr. Linda May. I think in order to be successful in this field, you need to be like Dr. May. It's not about how hard an individual works or about how, how many publications, although that helps. I mean, she is now number 7 in exercise and pregnancy, as it was recently published. But I think a really great deal is about mentorship. And I think that's what she really brought to the table for us. And I think a good mentor listens to those that are coming in under her. She built the framework of this study and then allowed all of us to really dive into and build a little part of it, a little arm of our own within her big grand study. I was able to fulfill my need of being able to still exercise train. As Bree mentioned, she wanted to get into that exercise again. I still was able to do the applied physiology stuff, and that was always important to me, is making sure that everything I do needs to be relatable to the actual person, right? I don't care about a downstream enzyme that may or may not matter, but I do look like the global metabolomics that Bree added on to my targeted, right? So we can actually see those different pathways and what's actually occurring, and that's going to be applied. So we kind of get the best of both worlds here. So yeah, I mean, is there anything you guys would like to add about any of this?
Yeah. Yeah. Linda's work is so impressive. You kind of pick your own path, and if you've got a question, usually it's there's data out there that you can analyze to address it. It's a good point. Bree, sorry.
I was just going to say, I think that you summed it up perfectly. The ability to have that framework and understand and listen to the people coming in to allow them to explore their own interests while it's still staying central to the point of having this really important intervention that is proving to be really great for maternal health and for infant health along the way, and just letting your interests take you wherever they want to go. It's a lot of fun, and I'm very grateful for Linda May for allowing us all the ability to come in and be a part of this team, but also have our independent interests emerge as well.
Yeah, and I think, oh, Alex, do you want to add something?
Nope, all you.
Okay. So I think maybe one more final thought from each of us, just for the students that are out there and kind of maybe feeling a little lost, because I remember my undergrad. That's why I have two bachelor's degrees. I couldn't decide, so I just continued with both, and I just kind of let the wind take me wherever. And that's, you know, like I mentioned, I made the mistake of sending a mass email. Right. That's, you live and you learn, right? But it actually worked out because I took a chance. And I think that's the biggest, my gonna takeaway from everything is always put yourself out there, take that risk, take that chance. The worst anyone's ever going to tell you is no. Then you're exactly where you're at before. So if you take that chance and take that risk, even if it is a mistake, you learn from it. But if it wasn't a mistake, you might end up in a program that you get a random email one day saying, "Hey, do you want to come to East Carolina and get a PhD working with babies?" I mean, I'll be honest, I was afraid of holding a baby by the time when I first entered the study. I didn't, they're so fragile and then everything. And then it was like one day I was in the lab with, or a baby visit with Linda, and she just, "Here you go." And I was like, "Oh, you're in it." So, I mean, again, I think you want to really just take a chance. It's really going to benefit you, and you never know what can come from even just the simplest email.
Yeah. Yeah.
Really.
Any final thoughts from either of you? Last
I
tidbit
will
of advice?
also
Go ahead.
I will also add on to taking that chance and, like Dr. Claiborne said, finding the thing that interests you. That was really my biggest takeaway through all of my schooling. I mentioned I was pre-physical therapy for almost my entire bachelor degree. And then decided that I couldn't picture myself doing it. And so I did feel that loss in my last year of, oh no, what do I do? And I just kind of listened to what was around me. I listened to a friend, I gave research a shot, and I've made my whole career out of it at this point. And so just staying true to you, because part of the journey is also the part of the destination is also the journey. And so you want to enjoy the journey while you're on it too.
Yep. Yeah, perfect segue, maybe without you even knowing it. I was just gonna say what Bree sort of finished up with. Be true to yourself. There is no right or wrong way. As long as you invest time and effort into whatever you're doing, you can kind of make your own impacts and be confident in that too, because not everyone's always going to agree with you. But yeah, just stick to your guns.
Well, alrighty. Well, once again, I am Dr. Cody Strom, and we have again Dr. Wisseman with us and Dr. Claiborne. I will kind of speak for all of them. If there's any questions that any of you watching may have, you are more than welcome to contact any of us. We're more than happy to talk about our paths, research, what we're doing now, whatever. But yeah, so with that, I think we're good. And, everyone take care. Thanks you guys again for joining me on this.
Thank you.
Thank you.
Thanks, everyone.